So let's just make sure I'm okay. All right. So thank you, Nick, for joining this session, and I hope that you have enjoyed the previous sessions through this research week. I guess Norman will be joining too. And I guess at the beginning of this, because I wanted to make this session a kind of interactive, at the start of it, I just wanted to hear from you. I'd love to hear from you about your thoughts about microscopic imaging, maybe medical imaging, how we do computational analysis using those types of data. If you can just tell me just one thing that you find interesting about what you have learned about this topic, and I would love to hear that from you.
Yeah, I think this has been a great experience so far. Just getting like a brief introduction to the computational microscopy and how it kind of works. I think it helps to like see the code behind it a little bit and the methods behind it. And then yesterday they went over like differentiating between different types of cells manually through the marking and then using that to train machine learning models. And I think that kind of put it all together, like what we're doing this for and why we're doing it. So I feel like I got a really good introduction so far to the.
Yeah. Yeah, that's great. And from your opinion, did you sense some challenge, or did you sense what are the important challenge? Maybe one or two challenge, maybe just one in this field particularly from what you've learned through this.
I
Um,
think what I sensed kind of was the qualitative kind of aspect of counting the cells and then like trying to have like a ground truth from the pathologist versus like training someone that doesn't have as much experience. So there's going to be like a dilemma there with what's accurate and not, and then using that to train a model could also create some inaccuracies in the model. So I think that was like the biggest challenge that I've noticed.
yeah, that's great. And actually, that's really true. So having this ground truth data that we can train our model on, given the style of the data we are having, is very challenging. And acquiring this data from pathologists, acquiring this data from expert or experienced um, personnel is really challenging and expensive, if we can say, because the nature of or the type of data that we are dealing with. And I guess to start our talk today is it's all about, if we see the medical imaging field and compare it to like medical imaging, and we have different types of images, and one of them is microscopy images. If you just compare microscopy images to radiology, or even to those optical acquired imaging in clinics, like those fundus imaging for the eye, and those things. If you just give like just a quick comparison, you can just notice something really distinguishable, which in microscopic images, we are dealing with large images, very large
Yeah.
images, because we are capturing it through microscopes, and the microscope can have very high magnification rate. So we can go down to the cellular level, we can go down even to the molecular level. So dealing with large images, that means that we have two aspects here. First aspect is, like, within the same image, if we have just one image, um, we need to navigate through this image. We need to do analysis. We need to do the artificial intelligence models. And those things are challenging within just one sample. And that's part of it. And the other part is, like, we are dealing with different, we're dealing with different levels, cellular level,
Right.
a molecular level. So we can get infinite number of samples. Imagine it. So if for instance, I wanted to capture the full resolution of everything of the cells down to the molecular, down to the DNA level, I can use electron microscopy. So, but that's impossible. So how I can navigate through the different biological level and get samples as much as I can. So that's one point. And also imagine the challenge of having this, if we can say, big data thing. Like if you think of it, just one sample of this, like perhaps one biopsy, one image of a biopsy in a pathology or histology images can be equivalent in size to perhaps 3,000 X-rays or 3,000 MRI images. So if I wanted to, and I don't need just one biopsy, I need many biopsies. I need to do sampling out of the specimen itself, So the storage of those images, the storage of those files actually is a very challenging thing. So, I mean, like, there are many aspects, there are many gaps that we can approach this field from. We can approach it from the engineering point of view, data engineering point of view, like the systems that are storing those files, the databases that are storing those data sets. And we can approach this from the data science perspective, like how we do analysis within just one sample of image. So this today, the practical part will be on one of our platforms, which is Digital Slide Archive. And Digital Slide Archive is a kind of a system that can deal with some of those challenges, dealing with large images. Perhaps you have come to that, but it's quite interesting to give you just idea about what is big images and how the data format is there. So if you see this satellite image, that's perhaps the satellite image of the United States. So that's a similar format. Actually, it's the same format that we are using with microscopic images, because if I want to go through this image, that's perhaps one image. So I want to see it at this level. But if I zoom in, I can zoom in very quickly. And at the end here, I see a very high resolution image, So in order to acquire those, in order to capture those kind of images, I need to, take photograph from the satellites and stitch those together in a certain way, and just combine those in a kind of format that I can navigate, go back and forth, user friendly, because without losing, the sense of, this quick and smooth navigation. So that's the same thing with microscopic images. So if I imagine that that's the same thing, I need to have this, if we can say, large image format, and I need to be able to navigate through this. And the way that this image is acquired, actually, you can see it here. There is a kind of stitching, marks here in this image. So the way this is acquired is as if we are taking a lot of images using microscope, and we are stitching those images to get together and storing them in a format that can enable this easy navigation and user-friendly, perspective.
So what is the resolution on these pictures? Like, how detailed are they?
Sure. So in terms of the size, so this specifically, it's around 100,000 pixel times 100,000 pixels. So this slide is like gigapixel resolution. So in terms of the spatial resolution of the pixel itself, like if I am just getting one pixel, just one pixel here, that's equivalent to a quarter micrometer in reality. So,
okay.
yeah, what you're seeing. So I mean, like for the size, that's something that might be, you know, perhaps I can show, if that's in person, I would have shown the whole slide to you, but imagine, um, so, so that's the slide.
Oh wow, okay.
Yeah, so if you know, because of this, perhaps let me turn off the background blurring so you can see it here,
Yeah.
that's how we store it in gigapixel resolution. So imagine the device that we use here, that's called whole slide image, whole slide scanner. All right, so the whole slide scanner is actually similar to the scanner that we use to scan papers, but instead of it does some sort of microscopic scanning. So it captured very small pieces of the image, tiles of the image, and it actually stitched those together. But there is a lot. That's a very sophisticated technology, because in scanner, um, the displacement of paper, stuff like that, that's very minimal. But here, through the mechanical vibration inside the whole slide scanner, this can the slide can vibrate. So there are a lot of techniques that help acquiring those images with very high resolution, and that's actually very sophisticated technologies. And to give you an idea how this is sophisticated, is that those technologies that used to do microscopic scanning of whole slide was there perhaps like years ago, but it wasn't until 2017 when those scanners was FDA approved to be used for clinical use. So
Wow.
because and even in 2017 it was just the first vendor, I guess Philips. I'm not sure. I'm not recalling this now, but it was in 2017 that it was having the first FDA approval for this device, and after that it goes, so we are now in this digital transition into the clinical domain. So most of our work yet still in the research domain, but there is like tremendous clinical transition towards this. So getting that said, that's just one image. So typical, perhaps laboratory in perhaps University of Florida, any medical school laboratory, they generate a lot of images per day, a lot of biopsies per day. So how we can store this in a storage and retrieve this, and how we design the processes, to go through all those images and to go through this, big data images that we are having. So at this point, I think I will start going through a little bit, like perhaps very basic, going through the digital slide archive. Did you have the chance to go through digital slide archive in the previous sessions?
I don't think so, no.
Yeah, yeah. So the digital slide archive. So I mean, like as highlighted here, dealing with just one image is a challenge. That a specific format that's stored in a typical pyramidal format. So in order to store this, we can store this using desktop programs, and we can also better store this in a cloud program that we can access from anywhere. Like that's our platform is one of those platforms that can be accessed through the web, and that can be accessed through through the cloud. So let me send you in the chat the credential that you can go ahead. I created an account for you for the sake of this research experience, let me, yeah, where is it? I guess, okay, let me, okay.
I was also wondering, do you have whole slide scanners at UF right now, or do you have
Yeah,
to slide?
absolutely. Absolutely. And I mean, like in we have whole slide scanners in UF. Um, Back in my old universities, I got my PhD in another university. They have multiple whole slide scanners as well
Okay.
at university level. So, but the thing is, and that's the trickiest part of it. So, the a laboratory in a hospital is an operational thing. So there are pathologists, there are residents that are, examining, slides. There are slides that need to be assessed in urgent way. So perhaps experienced pathologists can be on call, so they can, get to the hospital during, after hours, in case of, those who can say uh, transplant operation, those deceased donor transplant surgical procedures, the people that they are, got deceased and they were on the donor list. So, they need to do a quick pathological assessment, histological assessment on the specimen on the spot. So it's a huge thing, right? So until now, like perhaps I didn't witness that there is a lab that converted everything digitally. Until now, pathologists rely on this, slide, and they examine it under the microscope. And they are having those archive rooms, perhaps big rooms, and there are technicians that are like organizing the flow of physical slides, through the laboratory and through the processes, like scheduling those, organizing those, right? Uh, you know, Adding priorities and stuff like that to this kind of, this kind of operations. So what we wanted to do here is we need to um, make digital transformation for the whole process. Making a digital transformation for the whole process is something not trivial, because you wanted to make sure that you have system capability to store that, right? You need to make sure that, what about the outage, the system outage? So if this digital slide archive that we are building here, that's for research purpose. What if it was down and there is, so that's a kind of thing, the backup, the data storage, a lot of considerations, a lot of engineering consideration, and also scientific consideration, the quality that need to be considered while dealing with that.
Okay.
So I mean, like having this digital transformation, what I'm saying, this topic is very, it's hot topic, because perhaps through uh, maybe the next decade, we can see something fully digital in the clinical domain, not just even research domain. But I feel that we are going very fast toward this. Like if we imagine um, the same transformation happened before, uh, when we talked about those radiology images. If you remember back in the days, but perhaps 20 years ago, we was having we're having those films that we have those x-rays on the, so there weren't those like digital viewing, but now that's very robust. So all thing is stored on the computers, all things stored in the cloud. So now we can say that the radiology imaging, that's a part of medical images, is completely transformed into the digital domain. But we are now dealing with a different type of images with a tremendous, different scale of things that we need to deal with. But I feel that maybe a decade from now, we we might not be seeing those, physical slides. So if you go through this URL I provided to you, and so you can go here to the collections. So that's the digital slide archive. So instead of having this small archive unit that we are using to store slides, we're having this digital slide archive that we store slides into folders. You won't be able to see the whole set of folders by your account, but you can go to perhaps the sample images. You can go to images, and you can see very sample images that you can see here. That's very small images. We can also a very large image, but that's the kind of thing that we can go through. So yeah, so let's go perhaps more deeper into the topic, which is, I mean, we started this like in the previous sessions because it's all about this. It's all about marking things. It's all about annotating things. So we do cell marking, we do cell annotations, we do cell mapping, perhaps, we have a lot of things that we wanted to go through, right? So the thing is here, it all starts with that we wanted to make annotations on those images. And I'll just open this simple image here, or perhaps let me just start to get one, perhaps of, yeah, perhaps this one. But you can use any image, as you see here. If you just wanted to do annotations, you can just click on Open in Histomics UI. Open in Histomics UI, that's a kind of an interface to this digital slide archive that you can go more deeper into the sample image that you are navigating through. So you can see here that you have access to the image, and you can, navigate freely through the image, and you can do kind of annotations. Like, so the basic thing that we wanted to see, that we wanted to quantify here, even before before mapping and before doing anything, we wanted to do cell counting. So we wanted to annotate cells, and those are the nuclei. So if we wanted, like, assume that we have infinite manual resources, and we wanted to, make sure that we are utilizing those resources, we can create manual annotations here, right? So you can click new, and you can assume that you are having, a simple layer, that's the nuclei, and you can add description for this layer to the user, and you say save, and you can do simple annotations on, the image. So you can use point. So that's simple, that you do kind of counting to the cells, right?
Yeah.
So you can do rectangle, ellipse, you can do circle, and you can do polygon. So if you wanted to perfectly annotate, nuclei, which is something that's not actually being done manually, but I mean, like, as a conceptual speaking, we can do that. But we
Yeah.
can do kind of different annotations here. We can um, annotate artifacts, arteries, um, ROIs. We can do a lot of things through uh, this, tool. And as you see here, the annotation will be stored as a kind of metadata with this image. So that's really nice, because if we have access to this cloud tool, we'll be able to do annotations and would be able to add things that can be used or perhaps reviewed by other people. Like perhaps you can do annotations, initial annotation as a resident, and after that you can send those annotations to experienced pathologists, for instance, to to work on or to give you a kind of review. So we have here the kind of basic annotation things. We can add different layers. We can perhaps make a kind of ROIs that we see specific observations. For instance, here we can see that's a kind of quality aspects here that we can a kind of observe here. We can label that like that's an artifact, right? That's a kind of things that we can work on those whole slide image. So are you able to go through one of the images through Histomics UI, and were you able to do like basic drawings on that?
Yeah. Do you have a particular image you want me to use?
Yeah, you can go to sample images and images, and you can see multiple images here. Like you can go, that's I guess the biggest one here. I mean, like, in order to do annotations in whole slide images, you wanted to make sure that you have a kind of domain knowledge about things that you're doing. So basically, even within the histopathology domain, the pathologists are kind of specialists. There are specialist pathologists in every domain. So there are renal pathologists, there are liver pathologists. So I mean, that's a kind of, I cannot claim that I have expertise in like pathology. Even for me, perhaps I'm dealing with digital pathology, converting things into digital and creating those tools and softwares that can deal with generic type of data in the field of digital pathology. But in order to make specific annotations, we need to have this domain knowledge, and that's really important thing to know. But I'm not sure even what is the organ of this slice. Perhaps I might have guesses, but I'm not sure what it is. But at least we are assuming that we are engineering user and we wanted just to create a kind of annotations here. You can just go and just try the annotation on this image and try even to annotate this capillary, and let me know if you
When I'm trying to create a new annotation, it keeps saying access denied. Like when I try to, yeah, I'm not
Uh,
there.
let me check that for you. Can you just give me okay. All right, so
Yeah, I have the same issue. Access was denied.
uh let's let me fix that. The sample images. All right. Uh, let me, okay. Yeah, is owner. Access control. Your login with Research Experience Week, right? You should
Yeah.
you should be owner to this annotations. That's really weird, but I can't. Let me make sure to like so okay. Okay, let me close this and can you have another try on this now? And after that, if not, I will manage to give you access on the image, but that's Research Experience Week. Okay, wait. Segmentation. Segmentation access control.
Yeah, I still got an access denied when I tried to do it again.
Okay. Yeah, I will research. Okay. So scan is owner. Okay. So save. Okay. So access control. You have access now to this. Access control. You have access to this. So I guess you perhaps have latest try, and if not, I should have given you now access to, you know, so when can still you have this problem, access control problem.
Yes.
Oh, yeah. So that is, mm, mm, okay. Collections, sample images, and Sonic UI. Okay. Yeah, yeah, you're right. I'm not giving access to this. Okay, sorry about this. So I will and and um, and, and, so if I log out and log in again, yes. Okay, so yeah, so it works now. So I will log in here from or I guess you can try now and it will be working, and I tested it. So sorry about that. Just please let me know if
Yeah, that's working now.
yeah. So yeah. So that's actually one I mean, like, one of the we experienced one of the challenges that we can have in real workflow, if you are having slides, that's okay. But if you are having digital slides and there is I mean, like, a pathologist that would be kind of interrupted. So that's a kind of things, that's a kind of challenges in terms of the administration of the system and adding permissions, stuff like that. So if you so I guess please let me know your experience with that. If you what do you feel? You have any questions at this point?
Is there a way to erase with the
To
polygon? Like once you draw it, can you erase it or remove it?
you can so you can go to the folder itself, like, so you can if you see it here, if you can go to the sample images and images, and you can see that in the images itself, you can see that we have three here, like um, as a super factory, so those are three annotations. You can go to the file, you can see the annotations here, and you can see who created this annotation. So that's the account who created this, and you can delete these annotations according to the permissions that you're having.
Okay.
Yeah. And you can also view here. So I see here someone have created these annotations, and yeah. So yeah, I mean, so Winbin, if you can use this research experience account for now, and I guess I had a a kind of quick fix, but I mean, like that's a kind of developmental platform. We are just exploring things. So eventually, when we get this into the production pipelines, it would be, you know, IT department that taking part of it, and also in software engineering department, and different levels of, administration, to make sure that the workflow is not interrupted. So moving from the developmental stage, or from the research stage, towards the clinical, real life clinical things, is something that, that requires a lot of things, and it requires a lot of consideration. So I guess that was great so far. I will just using this research, experience account. You can go. I gave you access to more folders here. You can go to. You can go to the mouse model data, and you can go to the kidney PAS ERR aging folder. And after that, you'll be able to, go through multiple kidney images. That's actually a kind of free section using PAS stain of renal tissue. And we can see that we have, in renal tissue, we have kind of functional tissue unit that can be distinguishable. And if we have annotations for those things, that's actually the glomerulus. And so because the renal tissue is way complex compared to other organs, we can see different complexities, in the tissue. So we can see here that if we can do this annotations, manual annotations for this glomeruli, and the pathologist and the professional user here would be able to determine the stage of a certain disease, to perform assessment for this tissue. So we always wanted to have these annotations, and we always wanted to uh, uh, have those stored in an organized way. Different one what can access that in a different ways. And but the challenge that we're seeing here now is the challenge of the effort that we wanted to perform this manual annotation. That's a very challenging thing, that and time consuming thing. So one of the options here, one of the great options here that we can go through is you can do analysis, and you can do automatic segmentation by the aid of AI, by the aid of artificial neural networks. So I'll go with you quickly through multiple annotations tool. I'll go quickly first to perform quick annotations for the nuclei. So if we go to analysis and histomics TK, SOHAS KC, histomics TK, and if you go to nuclei detection, you can see here on the left panel the analysis ROI, and you can draw a rectangle here over the image. And after that, you actually submit. And I will give you some tasks to work on actually on that. Perhaps after a while, a kind of um, real same task is not time consuming, but I thought of having a kind of working with this digital slide archive and having a quick analysis tool that would be very useful. But as you see here, um, the nuclei detection performed this nuclei detections for sure. There are some missings, a lot of enhancement that can be done, and that's the way it is. So we might having some sort of like idea about the cellularity, and the cellularity, which is the density of cells in a specific ROI in a specific region of interest in the tissue. And the cellularity is a kind of very important aspect across the field of digital pathology, because you know, in oncology, in cancer, the cancer behavior is that we are having a kind of proliferation for the cells. So the density, the cellular density changes across the different parts of the tissue. And we can use those automated tools to have kind of idea, because guess what? If I run this here, so it will miss, even if the algorithm is not accurate enough, it will miss like 20%. But if I ran it inside a, you know, a malignant tumor, the algorithm will also make a mess. But we can see that the variability between the different regions here, and that's why we rely on those, you know, automatic tools. The second thing that I wanted to go through really quick here is the histocloud segment WSI, and you can just use the default model here, and you can just hit submit. That's just one click tool, and so we saw that we first make kind of segmentation or detection for the nuclei, which is something simple. That's not a complex structure in the tissue. But moving forward, we wanted to do the same to make detection, and after that to make segmentation for those complex structure, organ-specific structure. So that's the glomeruli, that's organ-specific to kidney, and we wanted to, you know, make sure that we are having a kind of robust detection for that. So I can show you here that maybe if we edit a little bit, okay. Yeah, it started now. So can you see here the progress bar? So it started, so that's around 5%. So what we are seeing here is that the tool is running those artificial intelligence model that we have, you might have learned through the sessions of this week. So you learn at different technologies, different computational models that can do segmentation, that can do detection. What we are seeing here is we run this model on whole slide images, and we can see here the progress of that. You know, that's it's running on the tile level. So when it finishes, it will be able to, you know, display that glomeruli, and, you know, instead of having this manual annotation process, we can, you know, save time and do it automatically. And yeah, until it finishes, let's like, please let me know if you have any thoughts about that.
I had
And
a question about when you were doing the border and like for the segmentation where you had a like rectangle around it where you just like picked out the cells. So it doesn't count the cells that are on the border of the rectangle or like actually touch the border that you created. Or
for this one, right?
yeah,
Yeah, so actually, what we do is that we are applying a deep learning model, an artificial intelligence model that do whatever logic inside this model. So basically here we specify a specific ROI, which is a region of interest. We direct the software that say, hey, and instead of uh, detecting the cells across the whole slide, that's a very computationally time-consuming task. We want to
right.
just to run the algorithm inside this rectangle, inside this polygon, and so that's a kind of the interaction that we are designing with the tool. So we wanted to make sure that perhaps the pathologist wanted just to run this to have an idea that the pathologist might be picking two regions in in the slide and just ran this analysis to have an idea about the thing.
Okay.
Yeah. So finally, as you saw here, you can just notice that we have those glomeruli detected and segmented, in the image without manual annotation. And that's the beauty of having this artificial intelligence model running, and integrated through this cloud platform. And we saw that we wanted to have a replacement for those archives in the histology laboratories, right? We wanted to replace those through the digital transformation, the anticipated digital transformation that we are working through. So we wanted to make sure that we integrated the things that we are developing in series, not in series, in research, like we are having models, artificial neural networks, we are having different things that can do different tasks, such as like perhaps detection for uh, objects in the tissue. Perhaps those objects can be in renal tissue, they can be glomeruli. Perhaps in other specific application, we can make it like detecting a specific tumor in the tissue. We can design a model that detect the tumor and classify the tumor in terms of um, the the malignancy, in terms of the stage. So we have a lot of things. We have a lot of things that can be running in the research background, but the things that, or perhaps one of the challenges in the field of digital pathology, that we wanted to bring this upfront to the user, and that's why tools such as the platform we are using and the plugins that we are building on this platform is something that might be very important. So we like, so for this for this specific glomeruli thing, we might not be interested about the count, the density. We may be in perhaps studying specific disease such as hypertension, but at least now we can use this just as a detection application. So we detected the glomeruli, we can just extract a specific tile of image around each glomeruli, and we can serve this to the experienced pathologist and say, hey, I have extracted those glomeruli to you, and do your assessment without needing to go through the whole slide image. That's a hypothetical application that we might go through this. So do you have another question so far?
I don't think I have any questions right now. What kind of other tools would you see as like a promising thing to explore? Maybe like certain plugins that you think that aren't present right now that you'd like to see?
Actually, I mean, like, I have a lot of things, maybe in progress. I'll show you one of those in this session. I'll show you one of those things that use generative models to generate virtual slides. But I think that at this point, there is a lot of things that can be implemented. I mean, like, that's beyond the implementation capabilities of the research labs that are existing. So if you think of the ideas, there are like overflowing the ideas into compared to the capabilities of what we are going. So that's a kind of booming thing that we are dealing with. So I don't have something specific here, but today would go through two applications that I'll show you. Perhaps after I'll give you a kind of exercise, kind of practical exercise, and we'll have a kind of break for you to get this done. And after that, we can get back and go through two other applications. One of the applications that we can go through is that we do mapping between this histology images and the molecular domain. And I'll show you how this mapping is meaningful and perhaps important to different aspects. And the other applications is I'll show you like the problem of generating virtual stains similar to this. Like, so that's a not real image. That's a this kind of deep fake things. So how we integrate this deep fake applications into the digital slide archive. So we'll go through that. But I mean, like, if you have a question I mean, like, until this point of showing the manual annotations and the detection, that would be interesting. I feel that we did not cover the whole things. There are a lot of capabilities that we can work through. There are a lot of softwares, but I just give you a brief idea, and through the exercise you can go and explore more about the capabilities of that. So do you have any other question? I hope that that was
I don't think
helpful.
so right now. I thought the deep fake thing was pretty cool because I remember in the microbiology labs that I've been in, the staining can be really time consuming, and then you just pull it up and it's right there.
Exactly. And I'm like, I shared with you the same interest that that's a very interesting problem. But the challenge working with that is that we wanted to bring that into the user, and we wanted to make the user accept. I'm sure that we're not targeting that we replace the those manual staining with something virtual, but at least we can have some sort of direction. We can provide some sort of direction to the pathologist, you cannot go with the different stains, the different assays. You cannot do the full set of tests on the histology level, on the molecular level. You cannot go to everything and collect everything. You can have some sort of selectivity and sampling, sampling across the data and sampling across also the test or the data domain that we are acquiring in the processes and in the protocols, and that's why that's my own belief that the generative AI and the deepfake things won't replace that. It will be such a guidance to say, hey, if you wanted to like do additional stain, and if you wanted to do additional processing, you may think of that, or you may think of that. So it will give you idea about what would you expect in that domain using those deepfake. So for the pathologist to say, hey, yeah, I agree, let's explore this direction. But I don't think it would be kind of replacing things, because there is error. There are errors in those models, definitely, because we are just relying on, detecting the dependencies between the histology, between the morphology and the molecular domain. But we have a kind of partial dependency, not full dependency. So there are additional information in the molecular domain for sure that wouldn't be related to the histology, that wouldn't be related to that. Like, for instance, if I'm seeing that cell and that cell, those cells might be identical in terms of the shape, in terms of the morphology. But if I do, a single cell sequencing for this cell and a single cell sequencing for this cell, for instance, I might be seeing some sort of heterogeneity in the expression profiles, right? So these heterogeneities might be related, might be dependent somehow, partially, on the morphology. So the thing I'm saying here, that those deepfake models are learning from the dependencies between the different modalities, but the modalities, for sure, by nature, are not completely dependent. So there are errors. That's why the generative models would be guiding in terms of just directing the sampling out of the whole word that you are seeing here. That's what I'm envisioning for this. So yeah, thank you so much for mentioning this.
Right. Yeah, it seems like a really useful tool, at least. Definitely not like completely replace like the slides, but just for idea generating and exploring different options without diving too much time into something that might not pan out. It's pretty exciting.
Exactly. That's exactly how I envisioned it. And yeah, I'll go through the plugin that I've developed for those things over the DSA Digital Slide Archive, and also I'll go through one of the tools that it wasn't developed by me. It was developed by Sam Border. So he developed the mapping tool between the histology and the molecular domain. I'll go for that too. But perhaps before that, let's go to perhaps a break. But I mean, like, through this break, I will send you in the chat some practical exercise, and let's go through that now. So did you see now my
Yep, I see your message.
okay, yeah. So the first thing So you go to the URL, the gdc.cancer.gov. That's one of the NIH data portals that study cancer data sets. If you go to this URL, that's one of the very nice portals here. You can navigate through the data through multiple ways. You can go to the projects. You can see here the projects, and the site here is perhaps the organ that is extracted through, and there are multiple projects across organs and multiple projects specific to organs, and we see here the different data collected. So we see the cases, we see like perhaps that's a sequencing breeds, and that's you know, you can see different types of data. You can see clinical, and you can see biospecimen data. And biospecimen data is actually the data that we are maybe more focused on now. You can see sequencing data, but this sequencing data or genetic sequencing data, that's both sequencing. So it's not spatial omics data mostly, but I mean, like that's a nice portal that you can see different types of data organized into, you know, a single portal. You can, you know, explore different things like just you can differentiate between, you know, gender, you know, vital status. There are autopsies. There are a lot of studies across, you know, the samples that are explored, you know, across multiple consortium projects. And if you go here to the repository, you can see the primary side, the project, the data category, and the data type, and most importantly here, the data format. If you navigate through the data format, you'll see SVS, so that's the whole slide image. You can see here 30,000 whole slide images that you can go through. So if we just select one of them, let's select this one and see. Oh, yeah, so you can see it here. Okay, so depending on the study that was conducted, you know, there are a lot of information that can be seen here as well. So the task that I'm just requesting from you, that you can navigate through the whole slide images available on this web portal and select two SVS files and download the files into your local machine, and you can create a collection in the digital slide archive and a subfolder with your name in that collection. You can create any collection. And after that, you can upload the SVS files into the digital slide archive, and you can just run the nuclei segmentation on the whole slide image. I did not mean to be running on the whole slide image as a whole. You can just select an ROI and you run that, you know. So, and after that, you just report the results, and let's explore the results after we have this break.
Okay, perfect.
Yeah, sounds good. So, so what do you think about what time do we return back? So if you're gonna stop recording and we return back, camera, if you help us in that? Okay, so I mean, like, what do you suggest? We return at we have this session until 12, right? Until noon.
I think so, yeah.
Yeah. So I guess we can have 30-minute break with it, or what do you think?
30 minutes?
30 minutes? Good.
I think that'll work, yeah.
Yeah, yeah. Sounds good.
Okay, perfect.
Perfect. So we'll meet again at 10:45. 10:45. Okay.
Okay, sounds good.
Sounds good. Thank you. Thank you so much. wanted to organize the data that we are having and playing with it. And annotation is an aspect here. So let's go through. Let's see really quick if just if we get a CIMAP, if you are able to generate anything so far.
How did you change the file from the GZ to the SVS? I think that's like the main thing I don't know how to do.
Oh, so okay. Let me. So it's Windows can do that through the command line. Let me, if I have this file, I'm not sure if I have one. I need to download it, but you know, you can use those softwares like WinRAR, and you know, you can use a command line as well to extract type of files, different types of files. And if you're working with on macOS, you can also do that. I mean, like that's perhaps something can add up to the hassle, but for the time of this, I guess I can send you some sort of instructions, yeah, offline to go through this. But for now, you'll eventually be able to have those SVS and upload them in a way similar to this. Sorry, similar to this as you're seeing here. And let's even see how it goes. But perhaps until then, as you might have sensed now, is that we're having a lot of tremendous amount of data, and the data are available online. And you know, even each sample of data is very huge in terms of size. And in each sample, you can have kind of, if you can say, additional information that can be added to the imaging information, which are the annotations. And the annotations can be performed by experienced personal, like pathologists, which are the people that are experienced with those types of microscopic images. And we can have annotations that we can just draw manually through our digital slide archive, or even through different types of things, but we can easily do that through tools such as digital slide archive. So if we rely on the pathologist to make these annotations, for instance, perhaps pathologist is seeing something, seeing some biological information here, seeing some sort of abnormality, normality. So this annotation can be added manually through the web interface. And we have different levels of configuration, different levels of accessibility that can govern this. But at this second part of the session, we just wanted to go beyond that. Like here we see that we have those models that can be trained on those manual annotations. So for instance, if I have manual annotation that can indicate a specific cell type, and I use those manual annotations to train a segmentation model, I can get a segmentation model that can whatever segment anything, even in like segment natural images or regular images, or perhaps traffic images, whatever surveillance. We can use those computer vision techniques in order to do the segmentation over, you know, the whole slide image. For sure, we have limitation in terms of we cannot run the segmentation on the whole slide like that. We needed to deal with this pyramidal format at which we navigate through the layers and we go, so dealing with that, that's a kind of hassle. And I guess through the individual projects you saw what are the steps in terms of like extracting data and organizing data into like tiles, and how we do a kind of annotation for that. But at the end of it, we reach out to a specific annotation that we are having, and for that annotation that we are having here, we train a model, and the model would be able at least to generate the same annotations based on the knowledge that we're given, or the information that we're given to this to this annotation. So the question here is, what are the other types of data that can be augmented into this imaging data that might be useful? If I mean, like, if you have any thoughts about that, you can share. What do you think can be useful if we augmented that into those images?
What do you mean by like augmented into the images?
I mean augmented, not I mean, like, so in terms of added that, I mean, like, so this image is providing, so this image actually is perhaps for tissue. The tissue is sectioned into small sections, or like sheets, very thin sheets. And after the staining and pre-processing fixation, a lot of stuff like that, we reach out to these images that can be seen under the microscope, so we can have a quality observation for the microscopic environment here. So the microscopic environment, when we talk about it visually, we mean the shape of the cells. So we see here the shape of the cells. We see the cells. We see the nuclei, right? We see the cytoplasm. We see we see the extracellular matrix. So we have here an idea about the microscopic anatomy using those images. So basically, this field is called histology, and histology is actually a kind of, if we can say, microscopic anatomy. And the field of anatomy is concerned with the structures, mainly the structures, and how the biological systems are built, and how it composed of but we are seeing it on the microscopic level. So that's the anatomy of it. But we wanted to get further information, not only about the anatomy. We wanted to get further additional information. So what type of data that you might thinking to be kind of useful if we have an additional information here that might be very useful, that we can say, hey, now we know more about this cell. Now we know more about this part of the tissue. That's why what kind of, yeah.
I was thinking like I was looking around on the website when we were on the break, and there was like genomic data, so I think that would be
great. Yeah, yeah,
so.
exactly. That's really nice. And the genomic data, or I mean, like, that's a cell. Perhaps that's a cell. We see it here. That's a border of a cell. That's a cytoplasm nuclei, right? And inside the nuclei, we have the nucleic acid. And the nucleic acid, there is a genome. I mean, the genome is like the same for the whole cells of the body, but at least we have some part of the genome are expressing in this specific cell. So that's very useful information. So part of it, we wanted to know the genomic information, and we wanted to know also the expression information, and that's a kind of different thing. So for a specific person, there is the genome analysis that we can do it. We can do genome analysis, perhaps from cells extracted from like blood samples, or we can use blood smear, or can use whatever sample we are using here. But we can also get to the level that we know what are the genes that are active on that genome for this specific cell. So if we think about, yeah, we have genome and we have different genes, and there are specific genes that are active that are defining the specific role of this cell, that would be very great information to have, right? Because what we are seeing here is just the shapes. So that whatever stains that we are having here, even if I'm using here, like that's an H&E stain, hematoxin or Eisen stain. So we can see clear the morphology of the cells. But I have here something called the trichrome stain that we can see here, the signature of collagen fibers. That's a specific substance in the tissue. So I can play with the stains, but at least now all what I'm seeing now is a kind of morphology, is a kind of shapes, is a kind of structures. But the ultimate way here, or the ultimate goal here, that we wanted to see not the anatomy also, but also the physiology of what's going on. Are those cells are functioning good, or are normal or abnormal and stuff like that. So genetic information can be very useful on that. So moving toward this topic, I wanted to introduce to something really, it's a technology that was actually generating a kind of a breakthrough in the field, which is, it started with something called Visium, with a company called Connect Genomics. It wasn't actually a company, it was a lab in Europe, and they developed this methodology that would be able to make, if you can say, genetic tests overlaid spatially on the histology image. So think about it. You do a genetic test, you get some blood work and do genetic test. That's a kind of, you are doing it either on the genome level or on the, transcriptomic level, in the proteomic, whatever part of the gene expression pathway, but you are doing it on a bulk level. So you are getting a sample and you are applying this genetic sequencing on the whole bulk sample that you are extracting from the body. But by the introduction of this technology, started with Visium and the other technologies that are doing the same thing here. What we are doing actually is that we are dividing, assume that you are dividing the whole slide image into small parts and whatever small spatial regions. And in each region, you do gene analysis, or gene test by seeing what are the genes that are expressed. It's basically, you are doing it at the transcriptomic level. So you are doing mRNA sequencing, but on the slide level or on the histology. And the way they are doing it actually is, it's really interesting that they are having a kind of, instead of this glass slides that we are usually using for histology, they are having a kind of active glass that contain spots, and the spot actually is containing the kind of primer that captured the mRNAs. and you're doing it, those those active slides having a spots, and each spot, it contains it can capture different number of mRNAs. It can capture around like 20K mRNA, and after capturing that, would be able to overlay this information on the whole slide image. So imagine if I'm having this spots and I'm being capable of I'm being capable of generating this gene sequencing down to this spot level. That's something really interesting. But actually, the key part about this technology is not. I mean, like the key innovation part about this technology is it's not logic that we are making the sequencing over each spot, like differentiating those into the sequencing machine that we are using. That's not a kind of something logic that we can, or that's not efficient. But the way they are doing it is that they are designing a spatial barcode um, in the primer that they are using to capture the mRNAs, and actually they are mixing the whole mRNAs from the whole, from the whole slide, and they are doing bulk sequencing for the whole sample. But after that, as a post-processing, they are getting the spatial barcode again, and we use this spatial barcode. It's a barcode. Actually, it's a physical nucleic sequence. So we have AGCNT, right? So that's a kind of barcode that we use to do some sort of overlaying over the whole slide image. So we need something like this. If you see this, so we have now the slide. If you see, that's a whole slide image on the left-hand side. And we have annotations, and annotations, perhaps that's the pathology or the expert annotation. That's a human that's saying, hey, that's something, that's an ROI, and that's another ROI, and so on. But on the right-hand side here, we see the regions of the spots. And you see here, that's the barcode that we are using to map this genetic sequencing on this specific point. So by using this VZM technology, and VZM technology is a kind of like being for years, but I mean, like the other technologies that are more sophisticated, but VZM technology is saying, hey, in this spot, I perform gene sequencing for the mRNAs that I captured around this spot. So we can think of, like, we are having now a kind of idea that the cells around this spot somehow they are expressing with this profile. So here, for instance, I'm just providing the COL3A1. That's one of the genes that are related to the collagen generation and fibroblast, stuff like that. So here in this spot, this gene is expressed. We captured 4 mRNAs with this gene, and in that spot, that's 0 mRNA with this gene, and that's a kind of spatial mapping between the genomics data and the histology data. So we can have this kind of mapping overlaid on the same X. So imagine now we have a tool that would be able to, say, hey, I'm knowing the cell. I'm knowing what is this cell, what is the state of this cells, how this cell is functioning, how is this cell is working. Is it normal cell? Is it abnormal cell? Perhaps there is a specific gene profile that can be a biomarker for a specific like proliferation activity. that map to a specific part of the diagnosis of specific cancer. So that's the level that we are working on when mixing and fusing between those histology images and spatial omics, and in that case, that's a spatial transcriptomic data. So at this point, please let me know and stop me if you have any question. I encourage you to, stop me, because we'll go further. Everything clear? Perfect. So one of the tools that we are currently developing here is developed by Sam Border. Let me send you the link over the chat. So you have the link now. So you see here that, I'm making sequencing for around 20K genes in each spot, but you see that I'm having this numerically. What are the opportunities that I'm having to just visualize this tremendous amount of information? So the genes there are a lot of genes, so I can do it on, I can do a lot of analysis from the genetic point of view. I can do enrichment analysis. I can do a lot of things, but I wanted to have a way to overlay those gene expression data onto the histology image, and that's why we are developing those tools that do this kind of fusion between the histology and the spatial transcriptomic. So in this tool, on the right-hand side, we have this, panel. It just show the image, which is the whole slide image, but I'm just viewing kind of early demo for this. But in the production, we are having the the pyramidal viewing capability as well. And if we just click on a specific spot of the tissue, we can see here a heat map of the spatial transcriptomic data overlaid on this style of image. So we can draw heat map for a specific gene, and we can also draw heat map not only for specific gene, we can do it for specific, if I can say, specific group of genes that can indicate a specific functions. Like, for instance, I'm knowing that there is a group of genes that can indicate proximal tubule. So I can select that. If I can, that's a renal tissue. I can, having genes that indicate podocytes. So I'm having this, this heat map for this specific gene, but this, this heat map is overlaid on histology images, and you can, see how we can utilize the genetic information and the histology information in order to map those data. We're not doing any artificial intelligence until this point, but all what we are doing is we are mapping between the histology image and the additional information that we can have. We can have different histology staining, like histochemical staining. We can have immunohistochemical staining, IHC, and we can have also, if we can say it, that's the spatial transcriptomic data and the spatial omic data that we can overlay on the image. And you can see that that's really interesting that the experienced user will be able to navigate through the different, cell profiles, and also can navigate through, the cell type proportion, and also the cell state proportion that can be driven from the genetic information that we are having. So do you have any question? Were you able to go to this URL?
Yeah, the URL worked. I have a question about the graph at the bottom.
Sure.
Yeah, so what's the difference between cycling and then degenerative and reference on the bar graph to the right?
Yes, sure. So let's say in the field of if we wanted to classify the cells that I'm seeing here in the histology image, I can classify them into two things. I can classify them into different types. So the different types, that means the different functionalities that I am seeing here in the tissue. So if I'm dealing, for instance, with renal tissue, that's a renal tissue that we are having here. That's the glomeruli. So I'm having specific cell types in here, and that's the tubules that I'm having specific cell types in here. And the tubules, we have this, nephron structure or microscopic structure. So we have this, this exchange through the functionality of the nephron, so we have different functionality even across the tubules. So we can classify the cells that we are seeing here according to the type, according to the functionality. And that's why what we are seeing here, that's a tubule, that's a tubule, that's a tubule, that's a tubule. But if we go to the spatial transcriptomic data, we can say, hey, yes, that's a tubule, but that's a proximal tubule, and that's a distal tubule, because we see the profile of the different cell types. So that's for the cell type, but there are other studies that can be helpful to know not only the cell type, but also the cell state. So each cell has a cycle. So there it can be generated, and there is a specific cycle in the cell. So in the pathway of the renal cells, we can use the genetic information, the genetic expression, to classify the state at which we are seeing this proximal tubule. So on the left, that's a proximal tubule, that's a cell proportion that we are having, that's the cell types that we are having, or that are belonging to the proximal tubule group, but we can classify also them to that's a cycling state, that the generative, that's a reference, according to the state. And by doing this, we would be able to, do a kind of profiling to see if we can, rely on this information to do like diagnosis, to perform further analysis, and to help the user to perform his own analysis. But what we are claiming in this tool so far is that we are multiplexing the spatial transcriptomic data with this histology data, and we are using those genetic algorithm that just make summarization for the genetic information that we are having, for this transcriptomic information that we are having, and drag it down into, maybe it's aggregating them into, or segregating them into, cell type proportion and cell state proportion. And from this visualization would be able to, the expert would be able to say something that we will see the community how it goes with that. But yeah.
So is that cell state based on the genomic data, or is it more the tissue, like just structure itself?
So all this is based on the genomic data. So actually what we are having here is like, say, if I just clicked on specific point here, so I'm getting a tile around this point, and by chance, this was a glomerulus, right? So I'm seeing here a glomerulus, right? So our tool here will aggregate the whole spots, the whole spatial transcriptomic spots within this ROI. So what I'm seeing on the ROI here, that's a glomerulus, right? So I'm aggregating the whole spots. Imagine like this glomerulus is this one, right? So I'm having multiple spots here that intersect with this glomerulus, right? So what I'm doing here is just I'm aggregating those spots with a specific mathematical aggregation function, and after doing that, I'm just using a specific cell type analysis and cell state analysis in order to, have that into specific profiles within the ROI that I'm seeing here.
Okay, that makes sense.
Yeah, that's great. So, do you have any additional question at this point? All right. So, we added here some like additional functionalities. If you can see the silk card, so we added some like educational information. If we see here, like for instance, we know that this is a proximal tubule from the spatial from the spatial transcriptomic data that we are having, so we can just get graphics or like a kind of illustrations, medical illustration that can be useful to know the type of cells. And so this tool can be, at this point, with this visualization capability, can be used for educational purposes, right? And moreover, we also included some reference tables that we see here, how this cell type is within the whole hierarchy of cell types. So we see the kidney, we see the nephron, we see the nephron tubule, we see the proximal tubule, and we see that's the proximal convoluted tubule segment 1, and we see those cells. So we are providing additional information. That's why I'm calling augmentation, in terms of adding additional information that wasn't be available from only the histology, because just the histology is just providing a 2D view of the tissue. But by using this kind of additional information, that would be helpful to the user as well. And finally, the latest portion of this tool is that we can do some sort of analysis and clustering across the whole, the whole slide, by just also doing a kind of clustering based of that information. Like, instead of just having it in a pie chart, we can get the full information of those patches, and we can run a kind of dimensionality reduction algorithm, like Disney algorithm. So that's a Disney plot for the information that are captured through those ROIs. And we can see some interesting information, like the clustering algorithm are saying like, hey, those are, those are meaningful to a specific point of view and not. So that's a kind of analysis that we are targeting from this tool. And again, that's still work in progress. And what you're seeing is a kind of pre-production version of this tool. And this is gonna be integrated into the digital slide archive to be accessed within this slide archive and slide management platform that you are seeing. So, any questions so far? All is good. Great. So, we mentioned now that what we are having here initially is yeah, that's the histology image, that's the morphology or the microscopic anatomy of the tissue. And we saw that we are integrating that with additional type of data, additional modalities, perhaps other stains, and also genetic information. And we are making this mapping between, the cells that we are seeing here and, the other information or the additional information that we are having from different sources. So the next, the last topic actually today will be in terms of augmenting further on that, we are thinking of using those, generative models. So generative model, that means that we wanted to generate synthetic data, synthetic information, instead of creating real information, synthetic information. Like, think about the deep fake technologies that we are having now. So what I'm developing here, what I've developed here, actually, that's a not real image. That's a trichrome stained slide, but that's not real image. That's a deep fake generated image from this HNE. And we're exploring the opportunities of using this, not for diagnostic purposes, but for driving the direction across the whole, samples that we wanted to sample from the domain that we are having. So instead of just collecting data, doing this brute force collection as a strategy, we can utilize this, generative AI models towards that purpose. So perhaps before going to, I have a plugin over the digital slide archive that can generate this virtual image on the spot. And before going further with that, let's get you some sort of quick idea, perhaps in 10 minutes or so, about the possible role of generative models in biological modeling. So we were talking about like biological modeling, and what we wanted to have here is that, what is a model? So a model is something that can represent something else. So that's actually a model. Like, so we have a biological system, we have a biological environment, right? So we wanted to have a model for that. So that's that's a mechanical system, but that's a model for an arm for the prosthetic technologies and so on. So in the computational domain, because that's my specialty, computer informatics, so we can have different types of models. We can have numerical models, we can have mathematical models, machine learning and deep learning. And because like those deep fake models and generative models are mostly deep learning, let's just see what do you think the most challenging part about deep learning compared to other machine learning techniques? What do you think? Did you do you have any idea about what's the difference between deep learning and machine learning and AI?
Is deep learning the one that uses neural networks to figure out the algorithms?
Exactly. So when we talk about AI, machine learning, and deep learning, so AI is that we are just designing a program that behave intelligently. So we can do hard coding instructions to behave intelligently, but that can be an AI. But machine learning, we do learning process. So we wanted to design a program to have like to go through an algorithm, and the algorithm would be responsible of getting trained from a data set. But in machine learning, we can use just statistical training. So the machine can be trained only based on the statistics the machines see in the data set that we are having. But in deep learning is based on the artificial neural network. So we are relying on the same concept that we are using in our even biological system, in our nervous system. So we are building an artificial neural network, and we are designing the algorithm that makes this artificial neural network learn from data. So our system is based on the experience. So what's interesting about deep learning is that we start from the output. We don't care about the distributions. We don't care about things. We care only about the output. We wanted to like design an artificial neural network, and we can just initiate it with random parameters. So if this network is given X, which is the input, the output will be rubbish. All right. So that's rubbish input, rubbish out. So the output will be nothing. But the way that the artificial neural network is trained is that we learn from the loss. So we start from the output layer, we compute a loss between the prediction and the ground truth, and we do the back propagation in order to update this, parameters. And that's why in deep learning, we are going feed forward and back propagation. We are going many iterations. That's why we are having those epochs, and we are moving forward in order to, complete the training process. So the thing is that the most important aspect in deep learning here is that we wanted to have annotations. We wanted to have something that the model will start from at the end layer. And that's why, depending on the problems that we are having, we need some annotations. We need just like annotation on the whole image. That's for the classification problem. We need perhaps bounding boxes that just indicate the image, or we can do those sophisticated segmentation. But at the end, we wanted to have this segmentation in place in order to start working with this. And as you saw in the whole slide images, making annotations is very hard. We cannot rely on manual annotations. So that's the role of generative AI model, that we can use generative AI for two things. We can use generative AI to even generate weak annotations, and that's very useful. The weak annotation are very important for the model to train from. And the second thing is just to give us an idea about what are the things that you might expect from the next domain. So the virtual images can give you an idea and say, hey, yeah, you can go ahead and extract those images and get those images and so forth. So I won't be able to go through everything and theoretical. I mean, like there are different types of generative models. There are GANs, generative adversarial networks. There are different types of GAN models, but I will, and we can do a lot of experimental design based on the different types of models that we may use here, but I will just go through. Let's see. There. Okay. So if we go through the collections and go through the research experience week, we can just upload. Let me just upload here really quick. All right, let's see. I have it here. Oh, okay. Mm, 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 mm. Mm. Might be ready in a minute. So until I get this sorted out, do you have any like thoughts about perhaps generative AI?
I don't think so right now. I think it's really cool though.
Yeah. All right, so let's get this uploaded. All right, so I will upload one image of the liver tissue.
When you train that segmentation model in histogram on hypergator,
Okay.
so what's the training data? So you have the image
Mhm.
and then the annotation.
Yes. You should
In-
XML files. If you're running on Hyper,
Yes, so images in what format?
SVS usually, they can also be MDPI.
No, I have SVS,
Okay.
and then annotation is XML,
Yes,
and there's a naming convention that it should match.
yes, they should have the exact same name beside the
Extension.
Yeah.
Oh,
They should match that. So test.
So if we just look into this image, that's a H&E, but that's a liver tissue. And one of the important factors that we wanted to go through liver tissue is we compute the fibrosis or like the fibrous amount of tissue to assess the point among the spectrum of liver diseases. So that's a kind of usual assessment that's performed in liver tissue. And if we look here, if we open that, would be able to just go through
That's available on Aggregator.
Ahmed Nagla, virtual trichrome, latest in virtual trichrome, and you can use the ROI selection here to select perhaps an ROI that we wanted to apply this model on. So we can apply it on the whole slide image, but we can just apply it on a specific portion here, and we set run.
They're basically just two separate scripts. So you submit the training script. Yes.
But I'm not sure if it's on, let me see.
The eval script works on, it's time-based. It's not like training step-based. So it's like after 10 minutes or after 20 minutes, run a random eval patch and give us the.
So perhaps in this instance, we might having some like difficulties at this point, but let me just show you here. Okay, so let me show you here on this Asina one, but the that's a production version. That's a kind of thing that we are working with. So we can run it here. Perhaps also we can just select some sort of
Or there's a way
we can do here, and we select the ROI.
If
And I'll send you an account also on the production.
starts to increase or something like that.
And we can select the input model, and from the input model we go to the model. So that's actually the deep learning model that's already trained to perform this deep fake, application. And after that we do submit, and it should work. Let's see.
And test on the remaining one. So what's the
As you see here, it's working, it's running in the background.
Yes.
okay, yeah.
Shouldn't be taking much. I hope that it works. But
You run this.
yeah,
You can run, you can run the viz on.
The one sample.
That one sample that you held out, or you could get a completely a different slide. Usually there's a two or three, it's split into threes, so you would use a couple for eval,
Right.
couple for viz at the end.
And when you run this, it gives you what? What's the output?
It gives you the annotation file, the predicted annotation file.
And you can compare with the ground truth one.
Yep.
Whether they overlap or something. Okay, let me try.
Okay.
And how do you set up the tensor?
so let's
So you can
see
type in Hypergator on
if
demand.
we
Just go to Google. and type
okay
Hypergator.
running.
Okay.
It should be the first link.
All right,
Here.
so it should be successful now. See, if we go here to the scene folder, we'll see something that was just generated recently here. We just okay, sorry, because see it here. So That's the ROI that we have selected, and that's the virtual trichrome predicted by the algorithm. And there are like a lot of enhancement that can be done. We can think about how we overlay. I mean, like, because we can overlay annotation on image, but image over image, we can think of the way we can better visualize this. But that's something still work in progress. But as you see here, at least if the pathologist wanted to just get a specific ROI and just think about what is the what if like what is the expected appearance of the the trichrome scene, the other type of scene, or the other source of information or a type of information. You can just compare and say, hey, yeah, let's request a trichrome scene of the over the whole scene, a real trichrome scene over the whole slide, or just get another section and apply the trichrome scene on. So that's the way of directing, the application of directing the sampling process. And the other way of having it is that, as you see here, there is a regions that's actually the collagen fiber regions here is not clearly distinguishable from the background. In the trichrome, we can see it easily distinguishable with blue color compared to the background, which is red. That's a cytoplasm, So you can have some sort of facilitated segmentation capability. You can run just simple thresholding over the trichrome in order to assess the percentage of fibrosis and assess the tissue much easier. So that's the thing that I wanted to share with you guys about the thing. I will send you credential on Athena, the production one, to check on the virtual staining module or the plugin. But I'd love to hear from you about your feedback, since we have almost having just 2 minutes.
I just want to make sure that we'll have access to all this for the next couple of days, because I know we have to create like a poster tomorrow, and I think a lot of these figures would be useful in trying to explain like.
Sure. So what is the due date of this poster? I can just make sure that I'll let the accounts up and running until that.
I think as long as it's up and running for the end of the week, like maybe like Sunday.
Okay. Yeah. That would be absolutely fine. I mean, like even perhaps to next week or whatever. But I'm not sure, because we are running like different scripts to do with those users that we are dealing with. So I mean, like there are like temporary users, but I'd make sure at least to have this up and running like for a week or so. That would be nice.
Okay, perfect.
Yeah.
I don't think I have any other questions at the moment, but thank you for spending with us. This was really
absolutely. And if you have any further question, you can just send me email. I mean, like I'd love to hear from you, and that's really interesting to have this kind of interaction, and you to learn about your thoughts about those things that we are developing. Yeah.
Thank you.
Thank you so much. Bye-bye.
Okay, so we briefly went over elevator pitches a few days ago. So today you guys are going to actually create an elevator pitch, and we're going to go more into detail about some things that you should include within your elevator pitch. And then I'm going to give you all some time to work on an elevator pitch. So for Friday's meeting, you guys are going to be present, and at this meeting, you're going to actually give your elevator pitch during the Hive meeting. So we're just going to go through and make sure that you guys have a nice pitch. They set aside 5 minutes for you all to do the elevator pitch on the meeting on Friday. So you have a max of like 2.5 minutes each, but it's probably not going to take you guys that long, because generally an elevator pitch is like 30 to 60 seconds, but you guys have extra time if you do go over that. So the purpose of an elevator pitch is to give your audience a brief introduction, make a connection, or engage them into a conversation. So it's pretty much like an icebreaker, where you're introducing yourself to someone, telling them what you have to offer to initiate a conversation or connection with them. Generally, an elevator pitch will include your name, your school, if you're a student, or your company. If you are giving a professional elevator pitch, it'll give your professional title. So for instance, hi, my name is Kimira Frost. I am a research coordinator at the University of Florida. It will tell what you have to offer. So for you guys' instance, because you are interns and throughout this program, you've been learning different skills, you can basically tell them in your elevator pitch some of those things that you all have been doing, some of the skill sets you have, some of the programs that you are familiar with using, things like that. Also, what are your goals / purpose for this interaction? So your elevator pitch will be different depending on who your audience is. And I know we talked about that a few days ago, but we'll go more into detail. So if you are at a research conference, your elevator pitch would be different than if you were speaking to someone during a meeting, or if you were meeting someone for the first time. It would vary depending on your situation, and as well as the goals and the purpose for that specific interaction. Also, you want to introduce your research or what you are studying. So for your elevator pitch, because you're going to be giving this pitch at the Hive meeting, your elevator pitch is going to revolve around the Hive program, the things that you all have been doing throughout the week. You can include some things you have learned or some skills, as well as some research or professional goals that you have for yourself that you may not have been able to necessarily experience here, but those are some things you'll include in your pitch. And then another thing, so you want to be unique and memorable. What sets you apart? I know we discussed this also, but just to take it a little bit further, you don't want your elevator pitch to be cookie cutter and boring. You want, after you give your elevator pitch, the goal is to either create a connection or to engage in a conversation. So you always want to set up the conversation in a way that it makes it easy for that person to respond or communicate back with you. So maybe they'll have some questions about something that you said. Maybe they'll have experience or expertise that they can give you. So just being unique and being memorable helps keep that conversation going. I know we've all probably had a conversation where it was kind of very dull and flat, and then after it's like, okay, what do we say now? So we don't want that. We want something that engages our audience, makes them want to ask questions, make them want to continue the conversation. Do you all have any questions so far? So some tips for creating your elevator pitch would be short and to the point. So we don't want to ramble. We don't want to continue repeating the same things over and over. We want to create your main talking points, make sure you hit those points, and then, move on to the next point instead of like talking in circles. We want to be memorable. We discussed that previously. Tell them what you have to offer, whether that's skills, education, a product, anything like that. You want to present what it is that you have to offer. Also, the goal is to create a connection, which would include building a network or a clientele. So you want to set up the conversation, and the ending goal is to add someone to your network. So if you're at a research conference, that may include talking to someone and getting connected with them on LinkedIn, or that may include exchanging business cards so that you guys can send follow-up emails, or that may include, let's say you're presenting research and then part of your elevator pitch, you mentioned that you're interested in a specific type of research, but you haven't had that opportunity yet. If this person has that connection, or maybe they do that type of research already, that would be a great way to build your network and possibly work for them, intern for them. So the goal of an elevator pitch is to really build a lasting connection. Then you want to seal the deal. So sealing the deal is basically staying connected by exchanging some type of information that would allow that connection to persist. So again, that could be a business card, that could be resumes, that could be contact information, whether it's their email, their LinkedIn. The goal is to, you know, expand your network by creating that connection. And then you want to follow up generally within 24 hours via email or LinkedIn. So for example, if you're at a conference, there are going to be so many different interactions, so many different people giving elevator pitches and speeches. It's not possible to remember everyone that you've spoken to, every detail about them, their name, where they're from, what company they work for, what school they go to. So the great thing about, you know, one, sealing the deal, which is getting some type of follow-up information, is you want to actually follow up. And the purpose of the follow-up is to really seal in that connection. So if you're at a conference and you speak to 25 people throughout the day, because you're not going to remember all of those people, having that, you know, that way to connect with them. Or if you're the person that's going out, maybe you go to a job fair and you speak to 15 different companies and you give them your resume. You want to follow up by email or LinkedIn with the person that you connected with, so that they can make the connection and actually remember, oh, that's the person that I talked to from the University of Florida. Oh, I already have their business card. I already have them on LinkedIn. And then just having that email or that second confirmation about 24 hours later is what helps you solidify and just build that connection. Okay, so this is just a brief example I created, but ours are going to be more in depth. But this is just kind of like a quick little elevator pitch. Let's say you join a Zoom call and they're like, oh, everyone, introduce yourself. Hi, my name is Kimira Frost, and I am a senior at the University of Florida studying public health with a concentration in social and behavioral science. My professional goal is to attend medical school and serve underrepresented populations dealing with health disparities. I look forward to improving health outcomes and advocating for health equity. So that would just be something very quick. You told them your name, your classification, your major, what institution you're attending. You told them a professional goal and why that's a goal of yours. So that's just like a quick elevator pitch. But for the purposes of this program, we're going to create something that's more tailored to, it's going to include these things like your goals, school major, things like that. But you also want to include the different things that you have learned within the program. Maybe things that stood out to you. Maybe you got a new experience here that you didn't previously have. So let's say you had experience working with Python, but not MATLAB. That's something that you can mention. Or if, you know, you've never had an experience updating your resume or even creating an elevator pitch, those are things that you can mention. So we just want to go into a little more detail to give the Hive members who are going to be at the meeting a broader perspective on the different things that you've done within the program and your outlook on it so far. And then a few things that elevator pitches are used for. So elevator pitches are used at conferences and interviews, at workshops, any public interaction. Maybe you're just in passing. If you're at school or work, they're also used online, but those aren't the only places. And the objective is this session is to help you develop the skills on how to effectively communicate research to a public audience in an oral format. So again, your objective or your goal of your elevator pitch will be different depending on who your audience is, your goals, and the type of connection you're trying to make. So if you're speaking to the NIH, if you're speaking to Hive, if you're speaking to a research group, your elevator pitch would include more of things that are research-based because of your audience. But if you're speaking to your class, let's say it's the first day of school and you walk in and your professor is like, okay, everybody introduce yourself. Your quick little elevator pitch would probably just be your name, your major, maybe where you're from, and then a fun fact about you. So it'll vary and it'll depend based on who your audience is. And then activities. So the interns, which is you all, you're going to prepare an elevator speech for the Hive team meeting, and then 5 minutes total is allocated. So that gives you a max of 2.5 minutes per student. Your pitch probably won't be that long, but you guys have up to that amount of time. Okay, questions. Do you all have any questions so far before we get started with the elevator pitch, actually creating one?
Do you have any tips for like being memorable? I don't know what that would entail, like how you want to approach.
So I think different things can make you memorable. I think your energy and just how you're presenting the information, making sure that you're enthusiastic, making sure you're interested. Let me give you an example. Hi, my name is Kimara Frost. I'm a research coordinator at the University of Florida. Yeah, I do research and like I work under Dr. Strekalova and like I don't really know what else to say. Yeah, I kind of like my job. I'm just really here for, you know, whatever. So one, delivery and presentation is very, very big. The way you speak to someone will make them either one, want to listen to you, or be tuned out. I think just being confident, right? So these people don't know you. So you could literally tell them that you are a rocket scientist or an alien from outer space visiting, and they wouldn't know if it's like the truth or not. So I think just being confident in your presentation. Don't feel like let's say we practice your elevator pitch and you include certain facts. If on the spot you forget those things, that's okay. Don't get down on yourself or don't feel bad if you don't say everything, right? Because the goal of the elevator pitch is to initiate a conversation. If you do miss out on some things or don't say things, that actually gives your audience a chance to speak back to you and ask you questions, maybe something you didn't mention, or maybe something you have mentioned, but they're able to elaborate on it. So one, I would say the way you present, your confidence that you exude, and just, you know, mention some things that your audience would like to hear. So I think, was it yesterday when we talked about anticipating what your audience may want to know? So because this is a research-based internship and your elevator pitch is about research, you want to include those different facts and experiences and skills that you have that are research related, because you never know. You may meet someone and they say, oh, this student is interested in this, I could use them in my lab, or I could mentor them, or, you know, things like that. So just try to create those connections with them that way. Does that answer your question?
Yeah, that makes sense.
Okay, and then we're actually going to work on them. So I want you guys to like pull up something that you can type so you can kind of like start to practice your elevator speech, like write something out. And I'm going to give you guys some time to do that, and then we're going to go over them, and then I'm going to give you some tips. Let me see. Do you guys need me to pull up? Yeah, okay, I'm going to pull this up. What to include? Share it again. So you guys can just pull up. Okay, you guys can just pull up your Word document and begin to type. One, you want to start out with an introduction. So you want to greet your audience. Hi, hello, something, you know, to greet them. And then you want to introduce them to who you are. So that would be first name, last name. Because your students, you'll tell them what school you're currently attending and your classification. And then you can begin to brainstorm what are some things about your research experience that you would like them to know. What are some things that you learned throughout the research experience? What are some skills you may have acquired or already have that are relevant to research? You could
Is
talk about, oh,
information
you can go.
about the Hive program?
Hmm?
Is there any more information about the Hive program or what that is?
Yeah, I can send you all a link. So pretty much you have like organizations within organizations. So CIMAP is like Dr. Panaki's lab. So that was like the things that you guys do when you do like the cell mapping and integrated reliability, right? That's CIMAP. And then CIMAP is under HUBMAP, which is under Hive. So it's like Hive is the big group, and then it's HUBMAP, and then it's CIMAP. So the Hive group is going to be like a larger meeting. That's why I said, you know, you guys can mention some of the things that you learned, some of your interests. But I will send you I will send you all a link while you're working on that. While you're working on that, I will How's your elevator pitch coming along? Do you guys need help so far?
I was thinking you said like to give a LinkedIn or like contact information. Would you do that at like the end or like suggest like
Yes, so that's not something you will necessarily include in your elevator pitch. That's something that you kind of bring up after the fact. So because the initial elevator pitch is like to initiate a conversation, right? So you speak, you allow them to ask questions, them to speak, and then towards the end of the conversation, like in wrapping up, hi, I would love to stay connected with you all. Are you all on LinkedIn? Can I have your email? So that's kind of like towards the end of the conversation where you would ask for contact information. So you can ask if they have a LinkedIn. You can ask for their emails. If it's somebody that you really hit it off with, that you feel like you would love to like follow up with them, make sure that you have their information so that you can send them a follow-up email, which can consist of a copy of your resume. It can consist of, you know, I really enjoyed speaking with you at the HUD meeting. That's another thing about follow-up. You want to make sure you remind them where you met them at, because again, sometimes they're speaking with and meeting so many people, you're not going to remember everybody verbatim. So having, hi, my name is Nick. It was so great to speak with you at yesterday's HUD meeting. I would love to stay connected with you. I have attached a copy of my resume if you ever have any opportunities and would like, you know, would like some help on it, you know, just things like that. And then you should have already gotten like, you know, their LinkedIn or their email at the meeting.
Okay.
Let's see. So tomorrow. And don't forget, you all have your informational interviews today with Dr. Sarter. So that's a great point of contact for you all also.
Right, wasn't it yesterday?
Oh,
I
was
think
it
I saw-
yesterday?
Yeah, it actually
Okay,
didn't
how
show
did
up,
that go?
so
Did it go well?
no, it didn't show up. So
Was it supposed to be yesterday or today? I don't. I
Yes,
didn't.
let me double check because I thought it was supposed to be today.
right, it actually said yesterday, so I don't know.
Let's see. Yes, it's today at 3.
Okay, I'll show up then. I waited yesterday thinking that it was like yesterday, so
Okay, yes. If you check your calendar, your Outlook invite, you should have it. Well, yeah, it's today. Your interview is at 3, Norman, and then Nick, your interview will be at 3:20. And I actually need to edit this because but yeah, it'll be today. I'm trying to find something that I can send you all about.
I think I have like a draft pretty ready to go for the elevator pitch. If you want to like.
Okay. Norman, how is yours coming along? Do you need any help?
no, I haven't finished. I'm actually Maybe I just need maybe 2 more minutes just to finish.
Okay, that's fine. That's perfectly fine. Hmm, I'm trying to-
Is it pronounced HUBMAP? Is that the way you say it for the?
That's the way I say it. I don't know if it's like I don't know if it's the right way, but I've never been corrected, so I'm just gonna go with her now.
Okay.
I know there is a high site, but for some reason I can't find it. I'm looking. Okay. So pretty much. HUBMAP is the national program, and then Hive is the one of the HUBMAP programs.
And the one we've been working with, is that the CIMAP?
Yes.
Okay.
Uh-huh. And CIMAP is like a lab. So Dr. Panaki, who you guys are going to meet today, you were supposed to meet him on Monday, but he wasn't able to make it to the intro on Monday. But he's over the CIMAP lab. So the people who have been coming in, helping you guys with cell marking, those sessions that are highlighted go on our schedule. They're from his lab, CIMAP. And then CIMAP is a part of Hive. And then Hive is a part of HUBMAP. So HUBMAP is like the national program. So you guys are going to meet with Hive, which is like a step below HUBMAP. And that's going to be like the different members of the Hive program. And generally just those meetings are, everybody kind of comes to those meetings and presents what it is that they've been working on. So you'll see like Sam, Nick, you'll see Dr. Ahmed, Dr. Yulia, myself, and then some other members there also. And then everybody just pretty much presents the things that they've been working on at the Hive meetings. And that'll give you guys a chance to connect. How far are you along on your elevator pitches?
I think I should be fine now.
Okay. Okay. Who wants to go first and practice? I feel like I want to volunteer Norman, because Norman, I think you let Nick go first the last time we did something. Don't be shy. It's not like,
I'm not really shy, but I think I'm gonna try to read it just because I mean I don't really know it by head, so
oh
I don't know
yeah,
if
that's
that is
fine.
okay.
You guys are welcome to like read off of something right now, because you're just practicing. The more you do it though, like the more you practice, not just today, but like when you guys are done, you can go home and just kind of practice. It'll become easier. And again, don't feel pressured to say anything specific. Like if you forget something, don't like beat yourself up, because they're going to ask you guys questions. So anything they want to know that you guys haven't asked, or anything they want you to elaborate on, they'll definitely ask.
Okay, that sounds good. Let me read what I have.
Okay.
Okay, so hello everyone. My name is Norman Janika. I am currently pursuing degrees in both computer science and bioengineering at Clear University. My passion is centered around harnessing my interdisciplinary knowledge to tackle pressing health challenges that affect our society today. My research journey has taken me across various aspects of bioengineering, from wet lab work, where I focused on isolating novel bacteriophages for medical applications, to the development of diagnostic devices tailored for low to middle income countries. In the area of dry lab research, I've delved into designing deep learning models for the label-free classification of different cell types. These diverse research experiences have ultimately ignited my interest in digital pathology. Recently, I've been delving into the pioneering work conducted at the CIMAP lab, and I'm enthusiastic about gaining a deeper understanding of how each group within the Hive is contributing to the advancement in digital pathology. I'm thrilled to join this dynamic community and eagerly anticipate learning from all of you, as well as sharing my own insights and experiences. Thank you.
That was great. That was great. Like, I don't really have much. I feel like you hit the mark. You told us who you were. You told us what you were pursuing. You told us your research, and then you also mentioned CIMAP, which is great. And again, any questions that they have, they'll ask them. But that was really good. So what we're going to do is we're going to go to Nick. You're going to give your elevator pitch. I'm going to give you some feedback, and then if there's anything else that we need to like target and work on, we will do that. And if you guys have any question about like how the meetings go, or who's going to be there, or anything like that, I can answer those for you as well, just so that you feel prepared. But everybody's very nice, and this will be a great opportunity for you guys to network and just meet some people that do all types of things. Like some of them are MDs, some of them are PhDs, some of them have like years of research experience, some of them are published, some of them have books. So these are just great people for you guys to connect with and then stay connected with even once this experience is over. So good for you to go to the meeting.
Okay, so I guess I'll do mine. So, hi everybody. My name is Nick Sherwin, and I'm a third-year undergraduate student on a pre-medical track at the University of Florida, majoring in health science and also pursuing a certificate in artificial intelligence and public health and healthcare. I'm also part of the honors program within the College of Public Health and Health Professions, and I will be working on my senior thesis with Dr. Strekalova. In doing so, I will be assessing the needs and usability of the HUBMAP platform by developing an interview guide, collecting interview data, and completing qualitative data analysis. This week, I've really enjoyed getting more familiarized with the CIMAP platform, as well as the field of computational pathology in general. I'm eager to start working on this project and excited to hear more about what the Hive program is working on.
Good job. You guys did really good. And again, I'm going to say this 1,000 times. Don't be scared if you forget something or don't mention something, because they will ask you guys questions. Let me see. That was to the point. You guys introduced yourselves. You told us what you were working on. You told us your interest. You said that you're a pre-med. Normally you said what you were interested in. But what may they ask you? Do you guys have any specific research interests, like anything that you absolutely want to do in the research world?
For me, I'd say my interest would lay in AI. So I don't know. I know I already said that with the certificate, kind of, but I don't know if I'd like reiterate that.
You mentioned that you were pursuing a certificate, but what you can do is towards the end say that you're interested in having more AI experience. Sorry, I got distracted. But that's the one thing that you could include, is that you're interested in research that involves AI. And the reason why you guys want to make sure you tell them your specific interests, because a lot of these people have their hands in so many different fields of medicine, research, things like that. Or if they don't, they have a colleague that does. So just throwing out your interests is very helpful, because they can connect you with somebody who actually works on that research. And that's how networking works. So the world is not about what you know or who you know, it's about who knows you. So it doesn't matter if you know that, this person, oh my gosh, this person does all of this research. They need to know you, and they need to know that you're interested in this specific thing so that they can seek you out for those opportunities, right? So that's another reason why we want to make sure we throw out some things that we are interested in so that we can build those connections. And also we want to make sure that we follow up, at the end. And I'll make sure I say it too, ask them to share their contact information with you all so that you can stay connected with them once the program has ended. Do you guys have any other questions about elevator pitches? Or is there anything that you want me to go over from the presentation that you probably didn't grasp too much?
I don't think so at the moment.
Okay, if you guys do have any questions, make sure you ask. What else was I going to say? And yeah, just make sure you guys are confident when you give your pitch. Don't be afraid. Everybody is super nice, super friendly, always willing to help you guys out. And because you are undergrad, you're going to be around some people who really have like a strong grasp in the research world, so they can be an amazing resource for you all to get involved in any way that you are interested in. Okay, so the final task that we're going to have today is going to be a reflection essay. So I'm going to send you all a template, and pretty much your reflection essay is going to be, so you guys know how I email you like every day and ask you to give us feedback on like the program. That's pretty much what it's going to be, but not just feedback on the program. You're also going to make sure you include, like reflect back on the things that you've done throughout the program. Tell us, you know, how they've helped you. I'm going to email you all a document, and the document is going to tell you what to include. Okay, if you guys check your emails, you'll see the paper, the document, and it'll give you the instructions on your essay. Let's see. I'll give you guys about 30 minutes to work on it, and I'll check back in with you at the 30-minute mark to see how far along you guys have gotten, if you have any questions. If you have any questions before the 30-minute mark, just feel free to unmute yourself and I can answer those questions for you. But I'll still be here. I'm just gonna give you guys some time to complete those. Did you find the dot in your emails? Did it come through yet?
Yep, it came through.
Okay. Okay, so you guys have been able to finish your reflection essays as well as your elevator pitch. And that is all that I have for you all on today. Do you guys have any more questions or anything that you want to ask before we conclude our session for today?
Also, is it Norman that's going first for the?
Yes, so Norman is going from 3 to 3:20, and then you're going to go from 3:20 to 3:40.
Okay,
And then after your informational interviews are over, you guys are good to go, but you guys will get 30 minutes back. So you can just spend that time preparing, figuring out if you have any specific questions for Dr. Sarter. And that's pretty much it. And then you'll be done for today, and then I'll see you all back tomorrow morning.
okay, sounds good.
Okay,
Sounds
if you
good.
guys,
Thank you.
you're welcome. If you guys have any questions, I'll still have my email up this afternoon, so you can just shoot me an email. Other than that, I will see you guys tomorrow morning.
Okay, see you tomorrow.
See you tomorrow.